Hello everyone, my name is Vitika Singh and today I'll be presenting my project on designing and validating an expression vector for GFP expression in E. coli using the PAT-DEST42 plasmid. In this presentation, I'll cover the reasons for choosing GFP, the cloning strategy, vector design, transformation, validation, and expression analysis. Moving on to our first Moving ahead, we have expression vector strategy. The slide outlines the key steps in our expression vector strategy for GFP expression in E. coli. First, PCR amplification of the GFP gene is performed using primers containing NDL and exol restriction sites, ensuring compatibility with the vector. Next, the PET-DES42 plasmid is digested with the same restriction enzymes, allowing for directional cloning. The GFP insert is then ligated into the vector and the recombinant plasmid is transformed into E. coli. Finally, successful transformants are selected using ampicillin resistance, ensuring only bacteria with the correct plasmid survive. Moving ahead, we have the plasmid map. So this is the plasmid map of PET-DEST42, which is designed for high-level protein expression in E. coli using the T7 promoter system. At the top we have the T7 lag promoter, which drives strong inducible expression of the inserted gene upon IPTG induction. The multiple cloning site, which is MCS, is located downstream flanked by NDL and exol restriction sites, which allow for directional insertion of the GFP gene. The vector also induces a C-terminal 6X histag, enabling easy purification of the expressed protein via nickel affinity chromatography. T7 terminator ensures proper transcription terminator for plasmid replication and maintenance. It contains an origin of replication and an ampicillin resistance gene for selection in E. coli. Overall, this vector is ideal for recombinant protein expression due to its strong promoter inducible system and purification tag. Moving to next slide. Cloning strategy and primer design. The GFP gene is amplified using PCR, incorporating restriction sites for cloning into PAT DEST42. The amplified fragment is then digested in NDL and exol, ensuring compatibility with the vector's MCS. Next, the digested GFP fragment is ligated into PET-DEST42 vector using T4DNA ligase, ensuring proper orientation of expression. The recombinant plasmid is then introduced into competent E. coli cells through heat shock transformation, followed by selection on LB agar plates containing ampicillin. For successful cloning, primers were designed to amplify GFP while introducing restriction sites for proper insertion. In the forward primer, we have NDL site, which ensures precise cloning into PET-DEST42. And for the reverse primer, we have exol site that facilitates proper vector ligation. Moving ahead to the conclusion and reference. So for conclusion, we have successfully cloned and expressed GFP in E. coli using PET-DEST42 vector. The system allows high-level expression under T7 promoter control and enables easy purification via 6X histag. Future work could involve optimizing expression conditions and testing GFP fusion constructs for protein localization studies. And thank you for listening. Here are my references, and I'm happy to take any questions from any of you.